This is Amir Sanati-Nezhad, founder and CTO of the Critical DX, or Critical Care Diagnostic. Criticare is developing lab-on-a-chip devices that is a combination of capillary microfluidics and electrochemical biosensors with a mission of delivering diagnostic devices that are combining precision, automation, digitalization, and miniaturization. When we started electrochemical sensors, we identified that those sensors has been designed, or electrode that were used for detection of those molecule or conditions, they were not designed specifically to quantify those molecules or measuring those concentrational molecule in very high precision. When we look at the literature and we notice that even working with electrochemical sensor, bringing that technology to the biosensor, so meaning that using that technology now for biomedical application and for diagnostic application, the challenge is significant because these electrodes have not been designed and developed for bioapplication. In the bioapplication, we are dealing with the serum. We are dealing with complex biofluidics that they have a lot of enzyme, they have a lot of complex molecule that they start interacting with the electrode itself. So we really engineered the screen printing method in order to fabricate new electrodes. And these new electrodes are specifically designed for biosensors, are specifically designed to be have a proper interaction with biofluidics or body fluidics. So when we look at the literature again, converting that electrode by itself to a biosensor is a combination of multiple steps, right? So you have electrode, you need to convert it to biosensor, which is able to detect certain molecule. So usually what happens, we add certain nanomaterial on the surface and we functionalize that surface and then you put that capture molecule on the top, and then eventually it's ready to detect certain molecule as a biosensor. But adding these multiple step materials on the electrode itself was another challenge. So not only the electrode was not good for biosensor, even the layers that we put on the electrode make the system pretty complex and also less reproducible, because reproducibility was very essential for biosensor. If you detect certain biosample from one person, if you measure 10 times, you want to make sure you measure 10 times the same concentration. That is the definition of reproducibility. But if you add these layers manually, one after another, or electrode, then you are not going to really have that reproducibility. So both of these challenges helping us or guide us toward engineering really the method of screen printing technology in order to have really stable electrode first, and second, eliminate all the layer of material that you need to deposit on the electrode. So we end up with really IP protected technology that is really this one. So these are like electrochemical sensor. It look like similar to conventional screen printing electrode, but the material and the fabrication process and the way that we engineer it is really specifically designed for biosensor. So now these days we call our electrode as a zero-step functionalization. So meaning that electrode by itself, once you add a capture molecule, is just converted to biosensor and is ready to be easily integrated into the microfluidic system. So we eliminated those multi-step deposition, which is really helping us to significantly improve the reproducibility of the sensor. It ended up with really demonstrated excellent for different diseases that we tested and we validated, and then that's what we are really using that technology for different diseases. Like a cancer, we are using for brain injuries, and recently we have used it successfully for COVID-19. The miniaturized one I show here, right? So that's the one, we have this electrode here. One of these electrode ones is easily as one of the layer of the chip is on the back, right? That's the same electrode that you saw there. Is already one of the layer of the microfluidic as a last layer. So once you integrate these two, already microfluidic is integrated with the electronic system. And that electrode is now specific for detection of certain molecule. As I said, you can detect RNA of the COVID-19, you can detect the nucleic acid protein or any of the target molecule of the SARS-CoV-2 or any other disease, as I mentioned, cancer and brain injuries and cardiovascular disease. So if you look at the combination of these function of that chip, its first is miniaturized, quite small, easy to handle, easy to store. And also it is really automated. So as I said, it's sample to result. You put a sample into the inlet and you just measure the signal from this electrode. Just plug into the readout system, which is normal conventional potentiostat, and then you have digital signal recorded. So it's already digital data. And it's highly precision, as I said. It's really detecting most of the molecule that we are detecting is a level of detection equivalent to the gold standard technique, and already perform much better than gold standard technique for quantification of those molecules. Yeah, gold standard technique for detection of protein, let's say, is ELISA. ELISA technique is very known for detection of protein. Our sensors has demonstrated to be 100 to 1,000 times more sensitive than ELISA on the clinical sample. We also have detected the RNA of the sample, which is now really equivalent to polymerase chain reaction, which is PCR, as everyone knows now in COVID condition, right? So that equivalence is already demonstrated on the clinical side, but we don't need to have bulky system. It's already miniaturized, it's already digital. We believe that this is the next version of technology can be used at home for monitoring your health, for precision medicine application, and for screening of chronic condition. So less need of the patient to go to hospital, right? Because now you can put your sample here, automatically the data and constitutional molecule remotely sent to the physician, and they can take decision based on that, right? So you don't need to they don't need to qualitatively evaluate you. They can quantitatively measure your health condition and then take decision based on quantitative numbers. So that is one exclusive technology, exceptional technology, we think, that is going to bring us from the lab now to the point of care, and now from point of care to the precision medicine concept. That is highly highlighted after COVID-19 situation.
Actually, the problem with all the products which are modified with nanomaterials is they are not reproducible enough, because the most important things in biosensing is repeatability and reproducibility, and they are so expensive. So we decided to have our own product.
So almost all of those that are commercialized, we already used those and we ended up with a challenge. Most of them, they need multi-layer steel coating, right? The electrode is electrode, but you need to add nanomaterial, you need to functionalize it, you need to add the capture molecule on the top. So we purchased most of those electrodes. They initially worked well in the lab, but when we start dealing with the clinical sample, we ended up with the challenge of either a lack of stability or lack of reproducibility, and kind of degradation sometimes happens. So there are a lot of challenges we are facing with that. Those are making system challenging particularly for clinical application. Yes, if you're going to use it for normal sensing, it's okay. Going to use it for really high precision diagnostic application, then none of those are going to work.
So all the sensors, to make biosensors from them, have the same problem. So one of the main problem with the sensors, carbon or gold electrodes, is we need to modify the surface with nanomaterial to have cross-linked material on the surface, to have biocapture molecule on the surface of the biosensor. So we decided to have this nanomaterial mixed into our ink. So we decided to mix a novel ink with carbon ink, and in this way we don't need to modify the surface of the electrode, and we don't need to have any cross-linker like NHS EDC or L-cysteine or other material to have biocapture molecule on the surface. We just need to immobilize antibody directly on the surface.
So we engineered this electrode, we ended up with highly reproducible. We increased the reproducibility from 10 to 15%, improve it to 2%, which is 2% is excellent for this kind of biosensor application. And we ended up with excellent clinical performance. So for COVID-19, as I mentioned, for cancer and for brain injuries, which are really our focus, but easily can be translated for any other disease condition. Some of those companies that they have commercial product, they start moving from one molecule sensing to 2 or 3 or 4 maximum. But when we are looking at the spectrum of the disease that we are talking about, brain injuries or neurodegenerative disease, or even COVID-19, if you're going to monitor your immune response, whether your body is immune to the vaccine or immune to the disease condition, we are dealing with more than 4 or 5 molecules. We are talking about really a panel of 6 to 10 molecules at the same time. If you are able to detect these 6 to 10 molecules at the same time, quantitatively, you already covered the entire spectrum of disease, right? Whether your disease is there first for diagnostic, whether your immune system is already involved in the disease, whether your recovery is on track or not. So that panel, that is the one highly useful for pharma company. They are the one interested to really monitor your health when you're using the drug, and it's going to really expedite the process of drug approval for those companies. So that's really moving toward precision therapy. Not just precision health and precision digital health monitoring, but precision therapy. The drug is designed specifically for you based on the biomarkers that we are measuring continuously from your health at home.